Thank you very much, Wolfram, and thank you very much, Jay, for having me here. Can you see my slides?
I
Yes.
can hear you good. Yeah, we can hear
Good.
you and we can see your slide. Yes,
Good.
it's
After thank you. After the very interesting presentation on COVID by Stephanie, I mean, my duty is to bring you back to the heart, to start by underlining that the loss of cardiomyocytes is really underlining most causes of heart failure. This is obvious after myocardial infarctions, we all know, but there is evidence that there is a loss of cardiomyocytes in virtually any cardiac disease and even during normal aging. And this obviously contrasts with the very limited capacity of regeneration. There are three types of evidence that the heart is a post-mitotic organ ranging back from thymidine analog incorporation to go through C14 DNA content analysis and the imaging mass spectrometry. Well, what is lacking in the field, in our view, is a way of tracing by genetic means replicating cardiomyocytes to visualize the rate of replication especially in mice. And so basically we set out to take advantage of 312 nucleotides in the promoter of the cyclin B2 promoter, which is cyclin B2 gene, which is activated exclusively at the G2M phase of the cell cycle. And we hook this as a short stretch upstream of the CRE recombinase, and then clone this into an AAV vector, and then deliver this AAV vector into ZEG mice, in which there is an inactive GFP protein which can get activated when there is LOXP recombination. And this gave us a sort of flavor of the rate of cardiomyocyte proliferation. This is a picture of the heart on the left side, in which we used CRE under the control of the constitutive CMV promoter. So this is an AV9 CRE injected in pups of ZEG mice. And so where you see in green means the cardiomyocyte that traveled through G2M and so underwent mitosis and gets stained in green and remains green for the rest of his life. So this is a cumulative way of seeing replication events. And these are other ZEG pups in which we injected the cyclin B controlling CRE. And basically, again, this gives an idea of the replication activity of cardiomyocytes in neonates. You see that there is not a very significant difference. The very big difference is when you use exactly the same vectors in adult mice. So this is a CMV CRE promoter. And you see that basically you transduce more than 50% of cardiomyocytes and there is expression, while the rate of transduction is the same for this AV9 here. However, CRE is not activated because cyclin B2 remains inactive except in very few cardiomyocytes. This gives you, I mean, a summary of this experiment. These are neonatal animals, and these are adult animals, and you see that basically the efficiency of this promoter is virtually null, and this reflects the replicative activity of cardiomyocytes. And the way to overcome this, we heard the different approaches during this presentation, some of which are very exciting in terms of a translational perspective. The way we thought to tackle this issue of regenerating cardiomyocytes is a very simple-minded one. It is to try and convince cardiomyocytes to re-enter the cell cycle. Zebrafish does, salamander does, and the neonate heart in mice and also in pigs does. So why they're not pushing cardiomyocytes to proliferate also in adulthoods? And obviously here the problem is how to find the right gene. And the approach we like to base our research on is not on educated guesses or on biochemistry, on pathway biology or omics analysis, but through selection of screenings. So basically we like to be as much systematic we can to do functional screenings and select what works. And it was upon this idea that we started a large series of screenings for siRNAs, drugs, microRNAs, anti-microRNAs to simply search for molecules that push cardiomyocytes into the cell cycle. And the most effective of these screenings are always those based on microRNAs, and we really fell in love with this category of genes. Our genome contains, in addition to 20,000 protein coding genes, the information to synthesize more than 2,000 different microRNAs. And this number means that these are many molecules, but not too many not to be screened in a single hit. So you can have 384 well plates, about 10 plates, you can screen the whole library of human microRNAs. I'm saying this in contrast, for example, of studying long coding RNAs. There are probably more than 100,000 of them, and not necessarily still well characterized. So in microRNAs, we know where they are, we can synthesize them, we can screen, they are small, and so they can be obtained in synthetic molecules. And in addition, they are pleiotropic, so each microRNA can target tens of hundreds of different genes, which at the beginning was seen as something that was a sort of promiscuous and undesirable, but instead is exactly what you want if you want to induce a complex pathway such like that of pushing a differentiated cardiomyocyte into proliferation. You need probably regulation of a complex pathway. So we started these screenings for microRNA and proliferation a few years ago. We identified a series that were very active in mice, rats, and human cardiomyocytes. This is an example of one of the most effective ones. This is a primate-specific microRNA 1825. Another one that is not present in mice and rats, but is very active in rats, as this example here. And the two most effective in rodents were Phy90 199A, and the genes coding for these two are delivered through an AAV vector, convert these big scars into much smaller scars with all the evidence of cardiomyocyte proliferation. We apply this method of visualizing by genetic means cardiomyocyte proliferation. We call this method cycle track because it's a method for tracking the cell cycle to these microRNA, and this is the result of this. This is 30 days after myocardial infarction in a control in which we injected the cycle track. You see some sporadic cardiomyocyte that replicate in infraborder zone. Instead, if you inject Phy90 or 199A, you see that 30 days after myocardial infarction, the number of cells that have traveled the cell cycle is much higher. What is even more interesting is that this becomes even higher after 60 days as a suggestive of a sort of ongoing process of cardiomyocyte entry into the cell cycle. This method labels cardiomyocytes to remain positively labeled throughout their life. So it's very different from VRDU that tends to be repaired from the cells. This gives you an idea of this regeneration. This is a big scar in a control, and this is the regenerative portion of the heart with this proliferating cardiomyocytes, for example, with 199A. A few words about these microRNAs. There are three categories of microRNA that push cardiomyocyte proliferation. The two of these categories are shown in this slide. One is represented by this large family of microRNAs that contain exactly the same sequence or the same sequence with an offset of 1 nucleotide. So the same sequence, which is instead of starting from 2 to 8 in the microRNA sequence, starts from 1 to 7 or from 3 to 9. And they're all these here in red and yellow. And these are very powerful in driving cardiomyocyte proliferation, but they also completely dedifferentiate the cells. So these are microRNAs that are very active in embryonic stem cells and tend to impart a phenotype of embryonic stem cells. The Ed Morrissey laboratory a few years ago made a transgenic mouse expressing 302, 367, and this mouse shows massive cardiac proliferation, but then dies of dilated cardiomyopathy because these cells don't differentiate. Another cluster is the 1792 cluster, is paralogs, which are shown here. And these are also powerful inducer of proliferation. However, they these microRNA are associated with cancer. 1792 was originally named the OncoMir1 cluster. And obviously for regenerative purposes, you don't want something that it can be associated with cancer. The third category instead is composed of sparse microRNAs. 1825 is one of these, 199 is another of those. And we like 199 because it is completely conserved in mouse, rats, pig, and humans. Very powerful, you can see here from this BRDU incorporation experiment. It is anti-oncogenic for thyroid papillary carcinoma, for osteosarcoma, for hepatocellular carcinoma. It is an inhibitor of the c-Met oncogene. And so this, we believe it is an additional safeguard profile. However, in cardiomyocytes, it is a very powerful inducer of proliferation. Over the years, we learned that the top 10 microRNAs in our list, including 199A, they all work on cardiomyocyte directly, so this has nothing to do with the stem cell story. And they work also on adult cardiomyocytes. These are Langendorf cardiomyocytes cultured with serum. They obviously don't proliferate. However, if you treat them with the majority of these microRNAs, they enter the cell cycle, incorporate BRDU, become positive for phosphohistone H3, and then undergo cell division by Aurora B kinase in mid bodies. And we know that there are two pathways for the majority of these, for all the microRNA, the 10 microRNA that we tested. And basically, one of the pathways is activation of the YAP transcriptional cofactor. If you silence YAP, basically none of these these microRNA work in driving proliferation. The mechanism of activation of YAP is different. 199A, for example, blocks beta-TRCP, which is an E3 ligase phosphorylated YAP. Others deactivate this inhibitory kinase, STK38L, and other ones, again, this kinase here, tau K1. So the mechanism is variegated. The final effect of YAP. This also means, this is also the reason why probably if we mix these microRNAs, we don't have an additive effect. The second pathway is to drive actin polymerization. And this is obtained by inhibiting a series of proteins that do the opposite. So drive depolymerization of actin towards a globular form. We wanted to see if this microRNA can work in large animal. This is a work team with Fabio Recca and his group in Pisa. We did the experiment in pigs, mimicking patients who undergo PCI, so coronary artery occlusion for 90 minutes, followed by reperfusion, and then intramyocardial injections of an A vector expressing these microRNAs. The results were really striking. They surprised even ourselves. So infarct size at 1 month was very significantly reduced. Cardiac function by MRI was very significantly increased. And there was a clear signs of remuscularization of these hearts you can see here, and you can see in the quantification of fibrosis. The treated are in black, and the controls are in white. My favorite movies, I always show this when I speak, is this one. This is an infarcted animal, 1 month in a control after infarction. So you see dilatation of the left ventricle, thinning of the septum, immobility of the anterior wall. And these are two animals which had the same size of infarct at 2 days, but were injected with an AV vector expressive 199A. The effect is quite evident. And there were all the correlates for myocardial passing through the cell cycle. This is shown here with phosphohistone H3 staining, so a marker of G2M. You see how many cardiomyocytes become positive for this marker. Now, obviously, you cannot use an AV vector for, thank you, for regeneration. For two reasons. First, because they express their transgenes indefinitely. A second, because it is unpredictable whether the cell will use the 5P or the 3P. And we know that in cardiomyocytes, for example, the 50% of the microRNA that is used is the 5P, which is undesirable. However, what you can do is to formulate the synthetic microRNA into a lipid nanocarrier. This is by far exactly the same that is now the formulation of the lipids are different, but the idea is the same. of the vaccines of Moderna and Pfizer BioNTech. So lipid nanoparticle, the one that we did, has a polymeric core with poloxamer PLGA, that coupled with the microRNA, condense it, surrounded by a layer of neutral lipids. These are the particles that we can obtain. They have approximately 120, 140 nanometer in diameter. We can inject them in vivo in the myocardium. These are mouse experiments. After 2 days, there is more than 100-fold than the control. Then the concentration progressively goes down, but it takes more than 10 days to go to basal levels, and a single injection in mice is sufficient to induce significant repair and preservation of cardiac function after up to 2 months from administration. What we still have to solve, and this is my last slide, is how to inject these nanoparticles in a large animal. And obviously we have the option of giving them intramyocardially and through mini thoracotomy. And we are testing the possibility of injecting them through the nova, inside again the myocardium and throughout the endocardium of the left ventricle. And we will also try injecting them, we are trying injecting them through the coronary artery, because nanoparticles of 120 nanometers could enter through the endothelial cells swelling, which becomes as large as 400, 500 nanometer. We like the idea of inducing regeneration by this, in this way, because we have a transient effect. Nanoparticles, as we know from the vaccine, can be stored in frozen vials, and they can be administered by an interventional cardiologist should the coronary injection be effective. So this work started in Trieste before I moved to London, is continuing by my group in London, and we have seen some of the names who did the work throughout my presentation, so we won't name anybody here, but I thank you very much for your kind attention and invitation to be part of this very, very exciting meeting. Thank you very much.